So sporophyte. Sporophyte is formed on the gametophyte in bryophytes. But how they are evolved, we are studying here, that is the evolution of sporophytes in bryophytes. To explain this, two different theories are there. First of all, what is sporogonium? That is the tissue which produces the spores. It is the sporophyte of bryophytes. So it consists of single terminal sporogonium with a bulbous foot. That is a primitive one, bulbous foot, and with or without an unbranched stalk or seta. So this is the primitive structure is explained. To explain the evolution of sporophyte, there are two theories. One is theory of progressive evolution. Second theory is known as theory of regressive evolution. So evolution of sporophyte by progressive sterilization of potential sporogenous tissue is called as theory of progressive evolution. Here what progress we want? More potential sporogenous tissue, more spore formation, illators, pseudo-illators, all these are not formed. I mean, the higher one, lower plant, they are formed. So, as we are moving to the higher bryophytes, there is in Polytrichum, Funaria, only spores are produced. All are potential spores. They can produce the plant body. So, this is a progress. This is progress of sporophyte. Sterile tissue is reduced, so it is called as theory of progressive evolution. Whereas second theory, that is regressive evolution. Evolution of sporophyte due to progressive reduction or simplification is called as theory of regressive evolution. This is about the theory. Theories we are calling, not that is not a, just these are the opinions, theoretical opinions of the scientist to explain the evolution of bryophytes. Theory of progressive evolution. This was supported by Bowers and Cavers and Campbell. So these are the three scientists. They supported the theory of progressive evolution, that is theory of sterilization. According to this theory, the sporophyte is a complex form. Complex form means Funaria, Sphagnum, Polytrichum, Pogonatum. In all this, they have evolved due to the progressive sterilization of the potential fertile tissue of the simple forms. Rixia is more simple, that is Rixia and Marcantia. Here the increasing, how they have increased progress, that we are simple to complex. Simple thing you can remember is simple to complex structure formation is theory of progressive evolution. The simplest form is Rixia and most complex form is Funaria or Polytrichum. So that can be explained under different stages. First stage, the simplest known sporophyte among the bryophyte is, that is Rixia. Rixia, it consists of only, that is sporophyte, there is only capsule, there is no food, there is no seta. So the simplest sporophyte in bryophyte is Rixia according to the theory of progressive evolution. Second stage is Carcinia. It belongs to the Hepaticopsida, the lower bryophytes only. Here in Carcinia, what you'll see is the small sterile foot-like structure you can see. The capsule has a single layer jacket wall. There is no seta. Only a small sterile foot is present and capsule is present. Capsule has a single layer jacket, but inside the capsule, some of the sporogenous cells, instead of forming spores, they develop into a sterile nutritive cells. They are also called as nurse cells because they provide nutrition to developing spores. Third stage, Further sterilization is seen in Spirocarpus, where the sporophyte has sterile bulbous foot and narrow seta. In third stage, you are seeing foot, seta, and capsule. In the diagram, you are seeing that Spirocarpus. So, Spirocarpus, you are seeing the foot, seta, small seta that is, and above that, you are seeing a round spherical structure that is a capsule. So, bulbous foot, narrow seta, and spherical, that is capsule is seen. This capsule, it consists of sterile cells, and it is sterile cells intermingled with tetraxospores. Spore mother cells here produces tetraxospores. Some of the spore mother cells will give rise to the sterile cells. Then, in turn, capsule, it consists of the layer that is jacket wall. Fourth stage, which is seen in Targenia. So in Targenia, the sporophyte consists of sterile bulbous foot and narrow seta, that which is also sterile, and a fertile capsule. Capsule you are seeing with, that is spores, that is spore tetraxospores, and elaters are also seen. So elaters like structures are seen in Carcinia. Then fifth stage, this is seen in Marcantia, where further sterilization of sporogenous tissue is seen when compared to the Torchinia. In Marcantia, the sterile tissue it consists of, Marcantia already we have studied, a foot, a broad foot is seen, a massive seta, and a single layer, that is single layered jacket wall of capsule you'll see, sterile apical cells at the apex of the capsule, and large number of long elaters with spiral thickenings. Some of the sporogenous cells will develop into the elaters with spiral thickenings. In this diagram, you are able to see different stages, that is evolution of sporophyte and bryophytes. First diagram, Rixia, which is very, very simple. A diagram, that is only spores. Then there is no food, there is no seta. Second one, Spirocarpus. There is a B diagram. You are seeing the food, seta, and capsule. In the capsule, you are seeing the spores and elaters. Then C is Torchinia. There you are seeing foot, very, very small seta, and a capsule with jacket wall, inside which spore mother cells and some sterile cells. You can see there is nurse cells. D, Marcantia, which is somewhat developed. In Marcantia, to protect the sporophytes, surrounding cells are also present, covering cells, and it will have a foot, multi-layered seta, and above that oval shaped capsule. Capsule, it consists of spores and elaters. Whereas the E diagram, what you are seeing is Pelia diagram, which is Pelia species, that is Epiphylla species. Here the specialty of this is Pelia is, you can see foot, elongated seta, and capsule somewhat round, and it is covered by the complete sporophyte is surrounded by a protective layer, that is calyptra like layer is wall is formed there. And speciality of this capsule is at the base of the capsule, inside the capsule at the base, you will see that is sterile structures. They are produced in groups. They are called as elatophores. So elatophores are present in Pelia, and above that you will see the spores and elaters. Sixth stage. So this stage is represented by some members of Jungermannians like Pelia, Ricardia. So here more sterilization of sporogenous tissue has been observed. Sporophyte is differentiated into food seta and capsule having multi-layered jacket. The sporogenous tissue produces mass of sterile elatophores and diffused elaters. Above the elatophores, you will see the elaters. Elatophores means 4, PHORE 4 means the stalk on which elaters are produced in the spilia. Seventh stage, that is Anthoceratopsida, anthoceros, which we have studied. The specialty of the anthoceros is here marked reduction in the sporogenous tissue has been noted in this anthoceros. Multi-layered capsule, it is differentiated epidermis with the stomata and chlorophyllus cells are present. So all the epidermis, chlorophyllus tissue, all these are developed here. So more sterile tissue is developed, that is developed here. Eighth stage, final stage, that is the members of the Bryopsida like Funaria polytrichum show highest degree of sterilization. The sporophyte is differentiated to food, seta, and capsule. The sterile tissue of the capsule, it consists of apophysis, operculum, that is capsule itself is differentiated in different parts, that is apophysis, theca, and operculum, like that. In that apophysis, sterile tissue of the capsule, it consists of apophysis, operculum, many layered jacket, and columella, which is a sterile tissue, trabeculae surrounding the sporogenous tissue, that is spore sacs surrounding inner trabeculae, outer trabeculae, and peristomial teeth. So all this complex structure developed. The sporogenous tissue is restricted to spore sacs only. So this is the final stage or the eighth stage, which is seen in polytrichum. This is about the anthoceros. And this is diagram of Funaria, where Funaria is somewhat similar to the polytrichum. Thus, the sporophyte of Rixia is the simplest among the bryophytes with a high portion of sterile tissue, and sterile tissue is very small. Whereas the members of the Bryopsida, that is Funaria polytrichum, complex one with all the complex tissue inside this. Sporogenous tissue is very, very small. The evolution of sporophyte and bryophytes as such is considered has taken place by progressive sterilization of fertile tissue. Fertile tissue is reduced here. Next one is theory of regressive evolution. Progressive evolution means sterile tissue is increased, fertile tissue is reduced. Regressive evolution, this theory it was supported by, it is regressive evolution is also known as retrogressive evolution. This theory was supported by Church, Kashyap, Gobel, and Evans. According to this theory, the simplest form of Rixia is the most advanced type. Here, just a reverse way. Rixia is more evolved. These people, they're supporting that. Rixia is more evolved. Funaria polytrichum is primitive type. Rixia, you'll see only spores. In Funaria polytrichum, spores are less. All other sterile structures are more. So according to this theory, simplest sporophyte of Rixia is, that is as compared to the simple capsule, it is the most advanced type which has been evolved by the simplification or progressive reduction of complex sporophyte masses, that is Funaria polytrichum pogonatum. Here the stages. Same in the reverse way, we have to explain. The semi-parasitic that is foliosporophyte, gradually lost its leaves. What is semi-parasitic? Foliosporophyte. Sporophyte, it consists of leaf-like structures. So that is covering is perigonial leaves, perichaetal leaves, all these are developed there. So they are all lost. So semi-parasitic folios sporophyte gradually lost its leaves and becomes embedded within the gametophyte. So directly on the gametophyte, capsule is developed, food seta are reduced there. Gradual reduction of photosynthetic tissue in the sporophyte. Third one is restriction or absence of stomata in only in the apophysis region. So in Funaria polytrichum, you will see stomata present only on the apophysis region. In sphagnum, the stomata of apophysis are not functional and they become rudimentary in sphagnum plant. Stomata are not functional. They'll not help in photosynthesis. Just they are present, but they are rudimentary. The capsule of most mosses, hornworts, that is mosses means Bryopsida, hornworts, Anthoceratopsida, and some Jungermanniales are multi-layered, which subsequently becomes single-layered. Multi-layered covering of the capsule is primitive, simple single-layered capsule presence of capsule wall, the jacket layer which is present in Marcantia and Rixia is advanced type, that is it is formed due to the progressive reduction of the layers. So this is progressive reduction. Stages of progressive reduction, among these changes there are gradual elimination of the seta and foot. So here Funaria polytrichum is primitive, Rixia marcantia they are advanced. So in Rixia there is no foot and seta. So gradually their foot and seta are lost. All these changes are accomplished by the progressive increase in the fertility of the sporogenous tissue. Sporogenous tissue is increased. Evidence from comparative morphology and experimental genetics supports the view that simple sporophyte of Rixia is not an advanced but a reduced structure. So, according to genetics and morphology, what they have compared is this is not the simple one, this is advanced, but why it is advanced means due to the structures are reduced here. So, this is about the theories of that is evolution of sporophyte in bryophytes. If you have any doubt, please you can ask.
Okay, my dear friends, regarding of today's class, Madam has clearly explained about the sporophytic evolution due to the
Lack of
very little time for our competitive examination. So, Madam has completed with bullet points and most crystal clear. So that's why I am very pleased to inform you. If you have any doubt regarding of this class, please you may ask our respected Madam. Otherwise, we're going to wind up the session. Okay, Madam, thank you very much. It's a great time. We are very happy, and it is a my privilege to have the most important valuable time for our spare your valuable time for our young aspirants, and also uh, you have more experienced botany fraternity in our Telangana, and also we are very lucky and thankful to you, madam, for spare your valuable time. Thank you very much.
Okay.
So I will going to
Thank you,
wind up the
sir.
session. Madam, thank you.
Okay, sir. Thank you. Thank you, sir.
Thank you.